Can everyone hear me? Hi, good evening, everyone. Happy New Year. I'm so glad to have everybody back. So we are going to continue our Science Cafe series here up in Saddlebrook with our series Mysteries Revealed, Discoveries in Chemistry with our partnership from the Chemistry and Biochemistry Department at the U of A. I just have a few quick announcements before I bring our amazing speaker up here. Just as a reminder, we do simulcast, live stream all of our talks. So all of our science cafes up at Chumamock, at Borderlands Brewing, Tucson Botanical Gardens, here, of course, on the Saddlebrook YouTube channel, and the Downtown Science Cafe at Magpies. All of you are familiar with the College of Science lecture series. That does start next week. They did give me all the promotion materials. That's why you see a minimal stack up front. Feel free to take as much as you like. I do know that they are doing the same kind of protocol as the years before. The lectures do start at 7, doors open at 6, arrive around 6:30 so you get a good seat, and then of course any overflow that's in Centennial is going to head over to social sciences. And of course you're going to have a bunch of people like me over there kind of leading the traffic if there is any overflow there. And Science City is coming up, and that is the U of A's College of Science and the Bio5 Institute partnership within the Tucson Festival of Books. So you'll get to see all of the different science that's happening across campus. It's 2 days, March 2nd and 3rd, Saturday and Sunday, from 9:30 to 5:30. If you have family coming in from spring break, this is a great time to bring them to campus, and it's a free event and open to the public, and there's activities for all ages. And I don't have that much for you guys. We're not doing much right now. Yeah, and of course, any information you have about or you're interested in about the chemistry and biochemistry department, I have all the materials up front. If you have any questions about any of the particular brochures up front from our different College of Science outreach venues, definitely let me know. And their contact information is also on there as well. Just a reminder, I'm going to introduce our speaker. He'll give his presentation. We'll do about 15 minutes of Q&A. And he did bring some really cool things to show you guys. So definitely if you're interested in coming up to the front after the presentation and asking questions, he has made himself available. And yeah, let's go ahead and introduce our speaker. So tonight we have Dr. Jacob Schwartz, a professor for the Chemistry and Biochemistry Department at the U of A. He received his Bachelor's of Science in Physics and his Master's in Science in Neuroscience from the University of North Texas. He continued with his PhD in Biophysics from the University of Texas Southwestern Medical Center and a postdoctoral training from the University of Colorado Boulder Lab of Comtech. He began his lab with the University of Arizona in August 2014, and he is currently studying, this is a mouthful, I had to practice this. He studies proteins that bind RNA and regulate gene expression with a focus on a specific group of proteins whose mutation causes a neurodegenerative diseases like ALS, childhood cancer, or even sarcoma. And then fun facts, like I mentioned, he did bring his fossil collection, and he's an avid fossil collector. He's also a volunteer foster for the Pima Animal Care Center. Tonight, Dr. Schwartz will discuss how life has always appeared magical to the human mind because it seems so well organized, and yet we notice so many things in the universe around us tend to decay. Biology at the level of the molecules involved has taught us that it is indeed the very tendency for all things to become more random and disorders that create all the beauty of the world around us and makes the amazing things that humans do possible. Please help me welcome Dr. Jacob Schwartz.
You guys hear me okay? Am I good? All right, great. Well, it's great for you guys to have me here tonight. It's nice to see such a great crowd. Yes, I'm one of the newer faculty at the University of Arizona in the College of Science and also in the College of Medicine, and I'm in the Department of Chemistry and Biochemistry. All right, let's see. There we go. It's going to work. So today I'm going to talk to you about how you probably are used to looking around you in nature and noticing that things are very well ordered. And you probably wonder sometimes, how can things be so ordered around you? Now I do love and have visited the Tucson Botanical Garden. This picture I happened to take when I lived in Fort Worth, so that's from the Fort Worth Botanical Garden. Here's an example of order. This is a beautiful Sidgwick garden in Kumamoto. This is backpacking with my dad. These are some lichens up at 12,500 feet that are still clinging to life under the remains of a glacial flow. This is looking down a spiral staircase at the Gaudi Cathedral in Barcelona, Spain. You can see again the order. This is not one of my pictures. This is from a museum in Southern California, but you guys probably recognize this as the symbol of life from the Tohono O'odham Nation. It's the man in the maze. And again, this is an example of order. I probably didn't even have to tell you the name for you to get the idea of what the story is about. However, though, life also has a tendency towards decay, and we know that, and that's part of our normal daily experience. If you've ever visited Berlin or Hiroshima, these two bombed out buildings have been left as a tribute to remind the heavy cost paid by war. This is again on Hawaii. I found this amusing that a lava flow had gone across a road, but it was cool enough, it left this sign just to let you know that the road was closed. If you have friends in Europe or family, if you get away from the tourist centers, you find all over Europe are dotted these castle ruins. It's kind of hard to imagine. That's the accumulation of someone or a whole family line's accomplishments sitting there collecting moss. Here in this picture is from Teton National Forest. Now some real jerk of a teenager carved his name and defiled this tree with his initials some 24 years ago, and the initials of some girl he had a crush on, who shall remain nameless because my wife's in the audience tonight. This is another example of decay. This is nearby in Yellowstone. This guy also had woman problems. He is the wolf who fell for a girl in a rival pack. He got away with visiting her about 5 or 6 times, and then her brothers caught him, and he didn't make it. So decay is part of something what we expect of nature. When you see something in nature, like a Mayan ruin, the remains of an adobe house out in Albuquerque, or petroglyphs here in Ruidoso or in Hawaii, you don't say to yourself, oh wow, that's random, that just came from nowhere. In fact, normally you would say to yourself, if you live here, you'd say, who did that? And if you have kids, you would say, when you find something in your house, who did that? And your kids don't ever seem to understand that that's a rhetorical question. They're not supposed to answer it, that it was a miracle. But in the case of life, in order to talk about where the organization of life comes from a biochemical perspective, we're going to have to break life up into its pieces. So this is one of my graduate students. This is Manta. And this biomass you see here, Manta, is about 3% Chihuahua. This is Hansel. He's one of our lab's pets. He's actually my wife's dog. He tolerates me. He loves to visit the lab though and see his friends, and he even lets me use his office where he receives his fans. Manta is made up of about 37 trillion human cells, so all of life is broken up into rooms that are called cells. The cell is the basic unit of life. Manta, not for any lack of tendency towards cleanliness, is also made up of more than 100 trillion bacteria. In fact, all of you are 3 to 4 times more of your cells are bacteria rather than human cells. That's because we all developed together and live in a symbiotic relationship. It's part of what makes you feel sick when you get too much antibiotics, is that you depend on these bacteria for your life. Now, whether you're a bacteria or a person or a chihuahua, you take away the water and you're left with about 3% of the mass is a molecule that you all probably all have heard of, DNA. You may have heard of the 20% of the mass is RNA, and 55% of the mass left over is going to be proteins. And so that's making up the bulk of the molecules that make up life. And these three molecules are very closely related to each other in what we call the central dogma of life. So, and that is that DNA has within it the code to make RNA, and RNA has within it then the code to make protein. And all life on Earth follows this rule. So this picture's, we're on Blackett's Ridge, so you can see a bigger chunk of Tucson. The Tucson area is one of the most ecologically diverse areas in the world, certainly the most diverse I've lived in. And all life follows this rule. DNA is transcribed to RNA, is translated into protein. So this actually happens in rooms in the cell. For complicated cells like yourselves, you are what we call eukaryotes, and that's because you have many little rooms in your cells. This one here is the nucleus. That's where the DNA is kept. It's highlighted blue here in this picture of these three cells. And so when the DNA is made into RNA, that happens in the nucleus, then the RNA gets transported out and into the cytoplasm, and then gets made, which is the outside part of the cell, and then gets made into protein, and that folds up into these beautiful, well-ordered forms. And so that's what our lab actually, we study the full spectrum. We are especially skilled in studying proteins that bind RNA. It's a smaller community of biochemists that will work with proteins that interact with RNA, even though these are, of course, the coolest molecules in the cell. But we study proteins that bind RNA, and they do so to trigger back and regulate gene expression at the level of the DNA. Now, I should point out, the way this information system works is, and you guys probably have heard of this before, these are all polymer chains. So these are chains of the same repeated subset of chemical components over and over again. So DNA, RNA, or protein are all polymer chains, and that's how they're read one into another. That's their basic chemistry. Now, for our lab, we're very interested in studying molecules that are, I get a lot of personal satisfaction out of studying and focusing on proteins that are involved in human disease. I could be studying slugs in the Amazon because I'm a professional scientist, but knowing that the work does double duty to help understand and hopefully come up with new therapies for human disease makes me happy. The proteins we work with, these are two very closely related proteins. I'm going to talk to you a lot today about FUS, and we also work with a closely related protein called TDP43. These are very important and causative of the disease Lou Gehrig's disease, or ALS. The Lou Gehrig family, actually, much of it lives here in the Tucson area. So y'all might be familiar with. This is what is, I think, rightly called a relentlessly progressive neurodegenerative disease. The motor neurons die. Life expectancy is 3 years or less. And there's no treatment that can slow or stop the progression of this disease. And that's because working out the proteins involved in this disease has taken a very long time, because this is one of the most complex, genetically complex diseases that science has uncovered. Now, in the case of truth is stranger than fiction, when I came to Arizona I decided to devote a separate part of my lab to study a disease that you probably would think couldn't be more different. Different mutations in the same proteins. So here's FUS and a very closely related protein called EWSR1, cause the second most common childhood bone cancer. So two diseases, bone cancer in children, a age-related neurodegenerative disease. But what I figured was we work on understanding how the molecules themselves work. So all our work to understand how these proteins work naturally, so we can come up with testable hypotheses to test in an ALS patient, would be just as important in these children with these bone cancers. And I have been rather impressed how well that has turned out to work since I've come to join the University of Arizona. Now, in getting back to that story I told you about, the dogma of life, so DNA is the information molecule. Proteins are the doer molecules. And the reason why they take up such a bigger chunk of the mass of your cells is because their chemistry is more diverse, so they've taken up most of the roles of doing the chemistry of life. Possibly, RNA can actually do both. RNA can be both an information molecule and do chemistry. So there's possibly a time in the past when these guys on either side weren't needed. But don't tell my RNA friends. We're going to focus on the ones on either side for tonight. So kind of skip over the RNA. It's a little more complicated. So you don't think I'm fooling you when I show you pictures of molecules. These here, I'm going to show you our conventions. These two are the same proteins. It's a nucleophore protein. And so this is just a representation where almost all the atoms are being shown, and as sticks is the connection, the covalent bonds connecting the atoms. Now, even Bill Montfort, who's another faculty in my department in chemistry and biochemistry, that's just down the hall from my office, who solved this structure, would agree that this looks like just a bunch of gobbledygook and hard to make heads or tails of. So what we tend to do is we trace out the backbone of that polymer, and this is called a ribbon diagram. And what we do is we also mark out with either these spring shapes or some of these ribbons here, certain features that tend to appear in proteins over and over again throughout life. So there are features that we see again, and you'll see those features again a little bit later. Now, to talk to you about how life achieves its order on the chemical level, I'm going to start using as a first example on the left side here, start with DNA. So DNA is, of course, you know why your children look like you. And as painful as it is to say, DNA is also why your children act like you. This is an example here of I've had 4 dogs of the same breed, Kubas. This is Junior. I didn't have a readily available picture of his mom, Shiva. They were both rescues. This is Chila. This is Chila and Junior when Chila's a little older. This is Chila when she was full grown. And this is my most recent Kubas I had. His name was Bator. He actually moved with me here to Arizona. And you can see that these animals all look very similar, and they share these traits of these curly coats and this white fur because they have very similar DNA. And my wife, she used, before she went back to school, she was the chief operations officer at the Pima Animal Care Center, and she's so embarrassed that her husband had purebred dogs rather than rescue dogs, and she had to confess to this. I told her purebred lives matter too. We've done both rescues and purebreds as well. Actually, it took almost better part of 2 years to find a responsible breeder for this breed. This alien-looking thing here, you can all agree is the same. This is another dog. These are all dogs. This is a Chihuahua. And these are all connected and look similar because of their shared DNA sequences. In fact, all of life is connected by shared DNA sequences. You maybe have not seen a talk before that talked about the central dogma, which is something that we think a lot about in the lab. You've probably seen talks before about evolution. The reason why Darwin is celebrated so much is because he drew this picture in his notebook some 60, 70 years before DNA and RNA was both discovered and rediscovered. So he really had no idea for a basis for why that is. Even in the 1990s, before the genomics era occurred, I don't think scientists had a realistic expectation of how much understanding the connections of evolution between different organisms would change how we understand human disease and how we develop therapies. If you go to the doctor, you are probably not going to be treated with a molecule that was developed to cure a disease in a person. Some people back in the 40s in one of those bombed out buildings I told you about did experiments like that on people. A doctor might prescribe a drug that was designed to cure cancer in a worm, but because we understand the genetic connections between you and a worm, then we can use the same, we can develop a molecule in the worm and use that to save lives for people. And that's why for today, I brought you another example of order found in nature. When I was in grad school, every weekend, me and the dogs would go to creeks and ponds and lakes around the Texas area and look for fossils in the Dallas-Fort Worth area. And Dallas-Fort Worth area is all Cretaceous limestone, so it's all in the 80 to 120 million years old. And the natural order you see in these fossils, I just always find amazing that they're so related. And particularly these are called ammonites. They survived for the better part of 300 million years and maintained this order. These are special. This one I like because it preserved that little hook that usually gets broken off when they get fossilized. This one is a more recent species that has a very fancy scale pattern on it. Carl Sagan has said that you are made of stuff of stars, and I told you that we work on curing diseases, understanding animal models. In my lab, we work with fruit flies and mice, human cells, and very generous donations from patient cells that were donated by ALS patients or children with sarcomas. But about the stuff made of stars, knowing that I have a piece of my DNA that I probably share with a T-Rex, I think is a little cooler than just being made of stuff of stars, but that's just me. All right, going to that DNA, let's talk about where this order comes from. You've seen this before. This is the double-stranded helix. Companies use this as their logo. In blue is the backbone of the two strands of the DNA, and what connects these DNAs, this is that simple. There's what I was telling you about. I said a polymer chain, and there's a simple code, just CGTA. And there's this pattern, so that's why these two strands are copies of each other. C is always with G, A is always with T, A with T, T with A, C with G. So these two guys are copies of each other. They can be used to repair each other. They can be used to reproduce, to make an offspring. And the thing that connects those are what are called hydrogen bonds. And in biochemistry, we'll look a little more closely at those. Those are among the most important types of chemical interactions that occur in molecules. So certain atoms, nitrogens and oxygens, are a little bit more loosey-goosey about sharing their electrons. So if one of those guys has a hydrogen, they're pretty okay with sharing that proton with another electron donor. So you have what form between them these hydrogen bonds between nitrogen. Carbon's not so good at sharing, so carbon doesn't usually do this. But these nitrogens and oxygens like to share hydrogens between them. Now, these you can see here, this is not ordered from disorder because these molecules are perfectly positioned for those hydrogen bonds to be in the right spot next to each other. So these covalent bonds are all properly placed to put those hydrogen bonds next to each other. They're complementary to each other. And how that happened actually is kind of funny. Scientists have argued about that for decades, and I probably shouldn't make fun of them because the chemists still debate about it. But about what the chemistry of early Earth must have been like in order to create molecules like this. Well, you're probably aware that the U of A has sent OSIRIS-REx out to an asteroid. It's actually reached the asteroid and is going to grab a pure sample and bring it back to us. It'll probably bring a lot of things, definitely bring a lot of things we don't know, but we kind of expect that what we'll find from rocks that have been brought back from the moon, a lot of the precursor molecules that are actually precursor carbon-based molecules for life. So it seems that making those types of chemicals is just natural to the chemistry of these atoms. And so all the debates about the early Earth chemistry looks like you don't even need a planet to get those chemistries to occur. Now, to go on to how to get the type of order that is made up of life, now I'm going to switch over to use for an example of proteins. So for proteins, these make beautiful structures. Every one of these structures were solved either at the U of A or by one of the faculty at the U of A. And so they make all these types of symmetrical and beautiful shapes that probably remind you of things that you've seen before. And they do this naturally. They're doing this all the time. They're doing this trillions of times in every cell of your body in every second. And so how does this happen? You're probably familiar with molecules that are in a solution are moving around randomly in a process called Brownian motion. So that this polymer that is a protein will spontaneously produce this beautiful structure seems very counterintuitive. So how does that happen? This is a protein that I like to use a lot in my lectures just because it's involved in your inflammatory response. It's called NF-kappa B, and it binds DNA and looks an awful lot like a butterfly. So these structures can be quite beautiful. One thing that's important about how life's order works is that it has to remain flexible. Life's molecules always have to be in motion. When they stop moving, they can't do anything. An example would be the amyloids that are associated with Alzheimer's disease. Those are proteins that have gotten into this very strange shape that they are completely immovable and nothing can bring them apart again. So they've lost their ability to do anything and become disease causing. So some randomness is important in order just to get in motion so life can be in movement and do its normal stuff. Now I'm going to try doing here and show you guys what normally we would present, I would teach on a senior level chemistry class or a graduate chemistry class. And I actually use the same slide before getting into this. So I've left the math on my following slides. But before you see the math, studies have shown that if you've seen pictures of puppies before you see math, you're more receptive, your brain is more receptive. That is scientific proof. Now, the path is so good at placing puppies so fast that they don't even get pictures of them before they find homes for them. So these are more puppies at heart, but hopefully it'll do the same for you. So this is a diagram that we use to add up the energy that goes into making a well-ordered protein structure. And here's a protein, and I'm showing you, letting you see here that this protein, even though it's well-ordered, it's still got movement in it. And that's really important, or else it couldn't do chemistry. So this looks really nasty, but we're going to go through this one step at a time. We'll start with talking about what the two sides of this are. On the left side, you have these negative energies. Now, by convention, negative energies we consider to be spontaneous chemical processes. So it just happens that we put a negative sign on it. So on the left side here are the forces that are driving life to be more, or protein to be more ordered. So like this cool spiral staircase. On the right-hand side, this is the penalty you're paying for making something that is more ordered. So this is, you have, this is called an entropic penalty. Entropy means disorder or disarray, and there's a rule that the universe always goes to more disorder and more disarray. Well, some of the energy that goes into making these proteins ordered are simple electric charges. Some subunits within a protein have just a simple positive or negative charge, and you all are probably familiar that positive and negatives attract. If you've ever stuck your finger in a light socket, you've experienced that. That makes up some of the energy. These are very powerful interactions, but probably rare to use possibly because they're too strong. And here's an example of such a charge interaction in an actual protein. Next to that is an unusual force called a van der Waals force. So this force comes from, every atom has a shell of electrons around it. And this shell is like a cloud. And just like a cloud, sometimes things move a little bit more to one side or to the other. So when you have a bunch of atoms packed closely together, like in a protein, you have an awful lot of atoms packed close together. These things can get into oscillation with each other and create just a momentarily partial charge on one side versus the other. This is a very small force, but when you have so many atoms, it adds up to a significant fraction of the force needed to bring a protein into a folded state. The bulk of the interactions are those hydrogen bonds that you've already seen before. So molecules that are involved in life, they are hydrogen bonds. Water, it loves to make hydrogen bonds with these same parts of proteins. Because water is just a hydrogen and two hydrogens and an oxygen. And you can exchange the interaction with water and have it become a hydrogen bond between two different parts of the protein. And this is also why water is called the lubricant of life, because then you can't undo that bond unless you had some water around to help take the place of that bond. So if you got rid of all the water, the molecule would be frozen and unable to move again. All right, so a lot of the energy comes from hydrogen bonds, but no, and again, we saw hydrogen bonds before when I showed you the DNA. And as also, when I didn't tell you what those spring shapes and those ribbon shapes are, those occur frequently in nature because they're held together by hydrogen bonds. The ribbons are held side by side together by perfectly oriented hydrogen bonds, or the coils also have perfectly aligned hydrogen bonds. And so that makes up a huge part of the energy necessary to fold a protein. But now, note, we're not quite there yet. We have not paid the penalty that is required in order to make a beautifully well-folded protein like this. There's a little bit of energy left. And also note that the indication here is delta S, and you see the same over here. These are all forces that are forces driven, much like you can think of the laws of gravity or charge collection. This delta S is a force made by disorder. So this is what is actually driving the last little bit to make a protein become well folded and make the order you see in life is actually a force that comes from disorder and from decay. We call this the hydrophobic effect. Hydrophobic is pretty simple to understand. As I said, this is a force that is a force of promoting decay. So hydrophobic, hydro means water, phobic means does not like water. I can assure you these cats were not at human animal care syndrome. This last little bit of force is because parts of the protein do not interact well with water. They have no polarity and no charge to them, so they just bounce right off of water molecules and tend to get pounded in towards the center of the protein. And then the parts of the protein that, say, have a charge on them tend to get oriented towards the outside because they like to be there hydrophilic, just like oil in water. Now, it's a little bit more complicated than that. In chemistry, when you have what's called a nonpolar or a hydrophobic molecule in solution, water has to make space for that molecule. So what the water does is it actually forms what we sometimes call a plaque. The water makes little hydrogen bonds with the other water molecules to make a well-ordered shell around that space for that hydrophobic molecule to occupy. And so when the protein gets folded, here I've just colored the hydrophobic bits blue, and here I put all the atoms in circles so you can see they're on the inside. Actually, all the water that was being trapped by being held around those hydrophobic bits is released. And so this process is actually a process of increasing entropy and increasing decay. And from that, you get a well-ordered and a well-folded protein. Now, for our lab, we study proteins that like to try to violate the rules. So we don't tend to talk about these proteins in the textbook. But there's not quite enough of a difference in energy to form such a nice, well-ordered structure like most of the proteins, like the 80–85% of the proteins that you would talk about in the scientific textbook. This is the dark matter of the proteomic world. We call these proteins intrinsically disordered because they won't form these shapes by themselves. They stay loose and open and flexible, adopt lots of different conformations. And here, what you're seeing here is Matt, the graduate student you saw earlier. This was one of her first purifications at the U of A, and you see the protein all stuck together to form what my chemistry colleagues call a big blob. So they say we do blobology, which I love Steve McQueen's movie, The Blob, when I was a kid, so I'll take that. So these proteins would just as much like to interact with each other as they would with themselves, and that's why they're not forming well-ordered structure. Why would nature do that? Well, it turns out nature is actually doing this to organize the cell, if you can believe it or not. What we've known in the cell, and it took us a while to realize, when I first saw this when I was still at the University of Colorado, I cursed the gods of science that I was given the worst protein in all of biology to work with. It took a while to realize, though, that in cells, this is a compartment of a cell. This is a nucleus like you saw before. The DNA is stained blue, and in green are colored these proteinaceous bodies. And these groups of proteins, and usually RNA, are known, and this is just one of many types that will spontaneously come together in a cell They don't have a wall around them. They don't have a membrane. These molecules just spontaneously form, and they're very liquid-like. They can disassemble just as fast as they're needed. And so this process, we realized that now for the first time we've been able to recreate this in the test tube. And so that's what we study in our lab. We usually don't study ones quite this big. That one Matta made was like a centimeter in diameter. We study very small ones that are in the microscope because we've also purified these out of cells, and we study how they interact with molecules that are involved in regulating gene expression. So we can go back and forth from the test tube to the cell. So what is actually inside this? Well, that is a matter of controversy. This is a very recent finding. If you looked at everyone's model, you as an audience member would agree with what one of my colleagues in Texas has characterized. He calls it the bowl of spaghetti model. Just looks like a bunch of spaghetti noodles clumped together. But what we've done in our lab, if we've taken that protein FUS, and this is our most recent publication, and we knew that it regulated transcription. We knew it liked to make those structures, but it had never been seen in a cell before. So we purified out from cells the transcription machinery, and this is our what we were expecting to find is that as transcription is occurring, a granule or an organelle, we call these granules, would form around the RNA. And what this does is it serves the role of organization. And we actually were able to find these. That little dot here, this is an electron microscopy, that's one of those enzymes. This is called a polymerase that transfers DNA and makes copies of it into RNA. And G is the biggest granule that we purified out of the cell that we observed in the microscope. It's pancaked onto the grid in the electron microscopy. It would probably be spherical shaped in solution. So this guy's about 250 nanometers in diameter. So why would you want such a thing in the cell? It's all about location. Do we have any former or current real estate agents in the audience? So if you have a hobby like sewing or carpentry, you need tools. You need needles, thread, scissors. You can solve your problems one of two ways. You can go to Amazon and fill your house with needles, threads, and scissors, so wherever you are, you will always have a needle, scissors, or thread. It's kind of actually, I do the same thing. My pockets are always filled with markers that I use for teaching, which my wife makes fun of me for that all the time. Or you could be organized, and like my mom has a sewing room, and she has a box, and all her needles and her thread and her scissors are very well organized, and they're where they need to be. And that's exactly what these granules do, is they serve the purpose of making sure life's chemistry is where it needs to be. This is where one you saw before. These are called nucleoli that are where certain RNA molecules called ribosomes are made in the nucleus. And this also, we believe, in fact, one person has gone so far as to call these processes the crucibles of disease. Because now this makes an interesting conundrum. These, now we know, are not proteins that are in disease forming these aggregates because that's something unusual. That's these proteins' normal function. They're supposed to be coming together in the cell and forming these organelles. And so Peter St. George Hislop has drawn this in one of his recent papers where he showed this would be the normal, reversible, almost liquid-like process of these proteins and RNA coming together. They're very dynamic. They can readily disassemble when they're not needed. But in disease, in patients that have either a combination of genetic mutations and possibly environmental factors, the structure of these are changed into an irreversible and non-functional state. And we've actually purified these out of samples that were donated by ALS patients and found indeed that these structures have been chemically changed and are no longer doing the function they normally have done. So this is a picture of actually, sometimes you can actually see these after autopsy in an ALS patient. These are those aggregates of that FUS protein that is not only aggregated together, it's not even in the right place of the cell. This is the nucleus, and the protein has left the nucleus and formed these aggregates outside of the cell. So this process gets hijacked. So that's how our lab is able to focus on these two different types of diseases. We think that there's two diseases, but one problem. Normally, these proteins' function is to help organize the nucleus, to help bring together DNA and gene expression machinery in where it needs to be turned off quickly. Life happens fast. When you get infected with a virus, your body needs to respond quickly, within hours. It doesn't have time for molecules to find where they need to be. They need to already be there. And so that's why the nucleus is a very highly organized organelle. And then in these, when you have these disease-causing mutations, or it can be mutations in other proteins that just cause proteins like FUS and TDP43 to make these aggregates, these are basically hijacking the system. So you're making these where they're not supposed to be, you're making them non-functional. And so we're studying an unusual case, which now I have to get used to the idea that we work with two diseases. When we think about what our discoveries means for an ALS patient, now we also have to think, what does the same thing mean for a childhood cancer? And when you think about how you engineer what you've discovered into a therapy, you have to think about the fact that what could be a therapy for one could actually be a cause of disease for the other, which both is good to know. You need to know the causes before you can develop a new therapy. But it's been an exciting ride to work on both of these two diseases together. So this is the people who actually do the work in my lab. This is Nata, you saw earlier. This is Lucia, Matt, and Rachel. Andres has just graduated and left the lab, and he is now at Rutgers University. We have had a number of undergraduate students actually work in the lab. The lab all got these t-shirts made for the Walk to Defeat ALS that was here in Tucson. It's every year. And this is Val. She's a staff scientist and actually one of the most gifted scientists I have ever worked with. And I also will thank our funding. So we're funded both by the U of A, but also by the National Institute of Health, the Neurological Disorders and Stroke Institute, and the Cancer Institute, and the American Cancer Society. And with that, I'd be happy to take your questions.
Go ahead.
I can run, I need cardio.
Yes.
What's used to the diagnosis of ALS? Is that a microscopic or biochemical testing?
Actually, that is Marta, who you saw her picture, that is the focus of her research project. Right now, it is diagnosed by observation. It takes a very long time. Obviously, you don't want to be wrong if you tell someone that they have ALS. So this can take 6 to 12 months. 6 to 12 months and the patient is not getting their treatment. And just with palliative care, a patient's quality of life can be raised so much more. And neurologists say that they could probably extend life by 2 or 3 years just by getting the palliative care early. We need biological markers. And so Matta is working on developing chemical markers because, in fact, I'm embarrassed to admit how long it took me, but being here at the U of A, we have both a med school and a university across the street from each other. So we were working on our chemistry, and I went by a neurologist and asked the question you just asked. You know, we're trying to come up with a high throughput assay to monitor this disease progression. Would that be useful to you guys? And she walked me through the experience a patient goes through of hearing the diagnostic of ALS, and I came away and realized, yes, this is very important. We're going to focus on this. So right now it is an observation. The neurologist, it's a metered observation. The neurologist will scale different motor abilities, but we're working on developing chemical techniques. Yes?
Anything new on forefront as far as MD and MS?
MD and MS. This is muscular dystrophy and multiple sclerosis. Yes, so for both of those diseases, muscular dystrophy has been having great successes on a new drug technique that's involved in also regulating gene expression. It's a technology I actually worked on in my PhD. They just got their first drug approved for spinal muscular atrophy. This is a version that occurs in children. And usually they die very young. So they're making strides in coming up with genetic treatments. As far as these types of diseases of aggregation, I'm a big believer that what we learn from one disease is going to readily roll over to the other. For instance, like Matta's diagnostic that she's working on, we're focused on ALS, but the same chemistry would work for your protein of choice. And then for multiple sclerosis, those problems are of an immune nature. There's ongoing research trying to, and that's kind of the forefront of research in neurodegenerative disease, is to understand the nature of the interaction between the nervous system and the immune system. They're normally not supposed to interact. That's kind of the point of having a blood-brain barrier between them. But I think actually you're correct in thinking that and a clinician would agree with you, as we improve our understanding of the immune system response to our effects on the nervous system, it won't just affect multiple sclerosis, but can also affect diseases like Alzheimer's disease and ALS as well. I think we got one in the back, and now I'll get right back to you. Yes?
That's exactly what I was going to ask you about. I just read somewhere where the lack of solubility of amyloid proteins in the brain is what causes the blockage and creates the Alzheimer's condition. And what I thought I heard you say was that it's pretty much the same thing in ALS because this protein becomes less active or inactive at all. Am I thinking right?
Yes, ALS has been pushing the boundaries of what we know because diseases, if you've heard of these diseases, Huntington's disease, you probably know of Parkinson's disease, Alzheimer's disease, all have these amyloid forms of inactive proteins. The aggregate forms that the aggregates we take out of ALS patients have kind of flown under the radar because they don't exactly fit the definition that was set by Alzheimer's disease. But I think at the end of the day, we're going to learn to open our minds and realize that nature is telling us that these types of structures forming are neurodegenerative, even if they don't quite fit in the narrow box of that's strictly defined by Alzheimer's disease. Now, there's a balance, and that's another area of study which the geneticists help us figure out, and us as biochemists, we worry less about because we're mechanism people. But the geneticists talk about gain of function, loss of function. So either the aggregate is doing something it wasn't doing before that is toxic, or the aggregate, that protein is no longer available to do what it was supposed to be doing before. As a mechanistic person, we often find these two to be two sides of the same coin. Because proteins can only do so many things, they obey certain rules. So I believe the answer is going to lie a little bit of both for the aggregates, that both themselves have some toxicity, but also is amplifying the toxicity by removing the proteins from what they're supposed to do. Oh, okay. Yes? Stephen Hawking is absolutely extraordinary. He does not actually hold the record, but less than I believe it's less than 8% of ALS patients live beyond 7 or 8 years. The life expectancy is 3 years with ALS. The Hawkins family has, in just the last couple of years, I think about 3 years, agreed with a lab in Southern California, a colleague, to have Stephen Hawking's genome sequenced in order to see if we can find the genetic cause of his ALS, specifically for that reason, to find if there's something unusual why he was able to survive so much longer than any other ALS patient. So it would be interesting to find out whether that is, it could go either way. It could be that there's a milder mutation that we hadn't discovered before, or it could be that there's an additional mutation that offset the disease and helped the cells compensate and allow life to continue for so long. I think I saw you first, yes. Yeah.
Did you say that there's no current medication that can be used?
There are 2 drugs. One was just approved last year for treatment of ALS. They extend life on the order of weeks, so they're not much of an improvement. Which is one of the things that drove us to realize the importance of a diagnostic. As you were asking me earlier, the neurologists believe that when an ALS patient has walked in the door, probably 70 to 80% of their neurons have already died, and you're not getting them back. So you need to diagnose this earlier. Neurons are able to compensate. They have a lot of plasticity, so they can keep the system going for a long time, and then it crashes all of a sudden. And when these clinical trials are starting, it's very late in the game to be hoping to try to help. So I think as we get better diagnostics, we'll start seeing very quickly more therapeutics become available. And yes? Yes, so mad cow disease drives proteins to form a similar structure that is an amyloid structure. They get called prions because they have the ability, without changing the DNA, so this is a way of transferring a genetic phenotype without changing any DNA. So these proteins interact with their partner proteins, and they push them in to form that same amyloid stacked sheet process, which is that crystalline form of proteins that there is no protein or enzyme in your body that can undo that fold. So there's theoretically, and there's people who believe, and I have no reason to disbelieve, that it is possible, given the right chemical conditions, you could force any protein into that energy well. We think of a spectrum of all the different energies. I showed you here when I showed you these arrows here. I showed you just a one-dimensional representation. Now, if you would imagine a three-dimensional landscape where that depth goes up and down over states. So for a disordered protein, it'd be very flat because the disordered protein doesn't much care what state it's in. For any protein that can form an amyloid, like the proteins in mad cow disease, there's a very deep energy well that life is unable to get out of. The only escape would be to find a way to eject them from the body because they cannot be degraded otherwise. So it is the same structure, it's a similar problem, and it's the same problem where one protein seeds another protein to keep that polymer growing with that amyloid structure. Similar in the same way that I guess another good example would be ALS patients. Again, we found that some of the ALS patients, most of the ALS patients we've studied, and most of the ALS patients that have a genetic cause known, they are heterozygous. That means they have one good copy and one bad copy. And yet, when we pull these aggregates out of the ALS patients, we actually pulled them out from donated skin samples. So we can even see the pathology throughout the body. We get both the wild type and the mutant form. So basically, the mutant form of the protein is poisoning the whole system and driving all of its friends and natural interactors into this same aggregate form. Does that answer your question?
Remember, quite a ways back that there was concern about how this disease may have and was related to
These diseases definitely, let's see, as a biochemist, I see these things, I see more similarity than differences than the clinicians see. The clinicians see very different specifications, and for diseases like dementia, Alzheimer's disease, ALS, the aggregates are forming in very different parts of the body. And so the symptoms agree with where those aggregates are forming in the body. However, as the biochemist, to us, it's a very similar process of the aggregates forming. Now, in the mad cow disease, that protein is able to interact with another protein like it so strongly and form these stacked sheets of proteins. And you're correct, just having the protein, so from eating the meat, the protein in your body will be driven into the same shape as that protein. And that is that indestructible amyloid shape, which can overwhelm the body. Actually, for mad cow disease, I will have to admit, I am not sure which part of the brain. So if a clinician was to argue why it's not the same thing as Alzheimer's or ALS, they would typically start with arguing about which part of the brain is affected. But on the molecular level, the mechanism is very, very similar.
Another question, I'm gonna.
Yes.
Is there any relationship or is any study of biotome versus biology?
The biodome, you said?
How the project you got.
Oh, yes. Absolutely, that is a hot area of research. So the question is, if I understand correctly, correct me if I'm wrong, those 100 trillion bacteria that live in your body, that they can be related to disease. Now, with ALS, a lot of clinicians believe that there's possibly all ALS has a genetic component. However, less than 10% of ALS is inherited through families. Most of it is sporadic. And it occurs so randomly, and it clearly clusters in particular groups of people. The military, the Department of Defense, is one of the great funding sources for ALS researchers, because soldiers are about twice the risk factor. Well, actually, soldiers of a certain era are twice the risk factor of developing ALS. So are athletes. It's not an accident that you see so many athletes on advertisements for ALS. They are actually at a higher risk. It's not just because they're athletes. So lifestyle or environmental factors must play a role. And the microbiome, we're actually just developing the tools to detect the differences. So I wouldn't be the least bit surprised if there's not an important component there. You've probably, I don't know if you've heard before, they can now, a colleague of mine who works on the microbiome, he's now at UC San Diego, has said he can tell you where you lived within 200 miles by taking a swab and measuring the bacteria up in your cheek. That's how quick. And it only takes 3 to 6 months for that change to occur. You move from somewhere to somewhere else, within 3 to 6 months, he can tell you again where your new location is. All right.
Any more questions? And just as a reminder, the next Science Cafe here at Sallenbrook is going to be February 7th. An additional professor for the Chemistry and Biochemistry Department, Dr. Thomas Gietti, is going to come. Evaluations are up front. Please feel free to all the materials, and I wish you all a good night. Thank you. 6:30. No, that's for the lecture series. I apologize. The lecture series for the entire College of Science is an overview. This one at Sallenbrook is at 6:30. Oh.